Hi, welcome to my presentation on microbial symbioses with humans. So there's actually a lot of different microbes living on and in your body. All of the microbes on your body and in it are part of what's called the human microbiome. There's actually a lot more cells in your microbiome than you have in your body that actually belong to you. So it's estimated that the average person would have about 10 times as many microbes inside and on their body as human cells. There's a lot of different organs in the body that have a microbiome. Basically, any organ that is exposed to the environment would have microbes living on or in it. And for any given organ, the microbes living in that organ are the microbiota of that organ. Or for any given body part, all of the microbes in or on that body part are the microbiota for that body part. So the microbiome is all of the microbes that you have as a whole. The microbiota would be all the microbes in a specific part of your body. In general, these microbes are good for you. At the very least, most of them don't harm you. So some of them are actively benefiting you, some of them are just not really affecting you. Sometimes you could have a pathogen in there that could potentially harm you, but that's not as common. So the microbiome is still an area of active research. We don't know that much about it. It's very complicated, so we're just kind of beginning to explore it. It is clear that it has significant impacts on human health, but we're not sure exactly what those impacts are, or I guess I should say like how you should alter the microbiome to get good health outcomes, to prevent disease or to cure disease. In the future, as we understand the microbiome better, we'll be able to manipulate it in ways that allow us to prevent certain diseases or to cure certain diseases. Studying the microbiome is pretty difficult, partly because you're working with human subjects and you have to always treat them ethically. So you're limited in the types of experiments that you can do because you can't do anything that would risk inflicting harm on the research subject when that subject is a person. So the main way that we've studied the microbiome so far is by taking samples from different parts of the body and doing genetic sequencing on all of the DNA in that sample, especially looking for rRNA sequences. So instead of actually culturing all of the microbes in your microbiome or in the microbiota for a particular organ, it's more likely to take a sample from that organ and sequence the rRNA in that sample, and then you can you can figure out which species of microbes are there based on their rRNA and how many species are there roughly. There's a lot that you can't know about the microbes in your microbiome, though, just from their rRNA sequences. So a lot of the studies that are underway now are trying to culture those microbes. Some of them are things that we already have cultured, like E. coli. It's the most cultured organism ever. But there's a lot of different microbes in the microbiome, and some of them have not been cultured. When you culture a microbe, then you can do tests on it to see what metabolism it does and to see how it interacts with different things, and what growth requirements it has. So culturing microbes is really important to actually having a strong understanding of their biology. It gives you much stronger understanding than just an rRNA sequence can do. In our explorations of the microbiome so far that have been based on rRNA, we've found that there's a lot of diversity in the microbiomes of different people. So there's certain species that are common to find in the microbiome, or certain orders or families that are common to find. But if you're just taking two random people, their microbiomes are probably very different in terms of which species are part of them and how abundant those species are. If you're looking at two people that belong to the same family, then there's a bit less diversity. So you tend to share a lot of the members of your microbiome with your relatives, and especially your close family members, the people you grew up with. See, the figure there is showing places on the body that currently are being studied. So you have the microbiota in those different body areas. Those are the subject of ongoing studies. So first we'll just go through the microbiota of all the organs that have one, starting with the gastrointestinal tract, or the GI tract. The first organ in the GI tract we'll talk about is the stomach. The stomach is an unusual organ because it has a really low pH. The pH around about 2, which is really acidic. The walls of the stomach are coated in mucus. It's a very thick layer of mucus coating the stomach walls to protect the stomach from all the acid in there. So when you're actually in the mucus, then the pH is not so extreme. It's still acidic, but it's not, it's not as bad. So you have microbes that can live in the stomach, in that stomach acid, and you have more microbes that can live in that really thick mucus coating the walls. Overall, not that many microbes live in the stomach. Because it has such a low pH, it actually has that low pH specifically to kill microbes. So you have some microbes that are adapted to live in an acidic environment, of course, and they'll be happy in the stomach. But most microbes that would be happy in your body will not like it in the stomach. They won't be able to survive there. Out of the microbes that are able to survive in the stomach, one of the most important ones is H. pylori, or Helicobacter pylori. It's estimated about 50% of all people on Earth have H. pylori as part of their stomach microbiota, and it is pathogenic. So it can cause a lot of different issues for the stomach, but basically it breaks down the mucus that lines the stomach walls, and then you have the acid of the stomach coming into contact with the walls of the stomach, and that creates tissue damage. So that causes stomach ulcers, and it can cause cancer as well, gastric cancer or stomach cancer. Most people who have H. pylori in their stomach don't have symptoms, so they're not going to be having like upset stomach and stuff related to the H. pylori, but it is still bad for it to be in there, and those people are still going to have an increased risk for ulcers and cancer later on. So just because you have the H. pylori in your stomach, it's not like a guarantee that you're going to get stomach ulcers or stomach cancer or anything like that, but it does increase your risk. After food passes through the stomach, next it enters the small intestine. You have 3 different segments of the small intestine. The first one is the duodenum, then you have the jejunum, and then you have the ileum is the last segment that leads into the large intestine. So the microbiota for each of those segments is going to be different. So starting with the duodenum, that is accepting contents directly coming from the stomach. So that includes food, but it also is going to include some of the stomach acid. That means that the duodenum also has a low pH. It's not as low as the stomach, but it's still low. So the microbiota for the duodenum is going to be more similar to the stomach microbiota. You're going to have more microbes in there that enjoy acid or at least tolerate acidic conditions. Then at the far end of the small intestine in the ileum, by the time you get there, the pH is neutral again. So it's like kind of normal body pH. That's also an anoxic environment with no oxygen in it, so it's going to support a different microbiota. So in the ileum, you have microbes that like a neutral pH and are generally going to be anaerobes. And they're usually actually clinging to the walls of the small intestine and kind of, sampling food as it passes by. So the small intestine is actually the part of the gastrointestinal tract where you're doing most of the digestion and most of the nutrient absorption. So the microbes that live in the ileum are actually kind of competing with you, with the walls of your small intestine to absorb nutrients. So some of the nutrients in your food are going to be absorbed across the wall of your small intestine. Some of them will be eaten by the microbiota of the small intestine. You have all types of metabolisms in there. Most of the microbes in the ileum are able to do a number of different types of metabolisms, or they're able to use different molecules as a source for energy. That way they can still survive even if your diet changes or something like that. So the microscope images there are showing those bacteria actually in the ileum of the small intestine, clinging to the walls of the small intestine. Then in between the duodenum and the ileum, you have the jejunum, and the jejunum is kind of like intermediate. It has a pH that's a bit low, but it's not very low. So the pH is going to be rising as you go from the duodenum to the ileum. So it's rising throughout the jejunum to up to a neutral level. So the microbiota for the jejunum is going to be kind of in between the duodenum and the ileum. It looks more like the duodenum on that side, looks more like the ileum on that side. And then this figure here is just kind of showing the organs in the gastrointestinal tract. So you start with the stomach on the top, and then you go into the small intestine. So the blue segment of the small intestine is the duodenum, and then this kind of orange segment is the jejunum with an intermediate microbiota, and then this purple segment is the ileum. And then wrapped around that is the large intestine. After food passes through the small intestine, it moves to the large intestine, which also has 3 different segments, and every segment has its own microbiota. But for the large intestine, we're only going to worry about one of those segments, the colon. So the first segment is the cecum. On this diagram of the intestines here, the cecum is really small. It's just right here. It's this tiny little part right where the small intestine merges into the large intestine. Then you have the colon, which is the largest part. All of this is the colon. And then the very last segment is the rectum. So we're only going to worry about the microbiota of the colon, which is by far the largest part of the large intestine. The colon is anoxic, so you have anaerobes in there. Some of them are actually facultative aerobes, so they're able to survive without oxygen, but could use it if it was around. And the colon is just really highly populated with microbes. So out of the entire gastrointestinal tract, most of the microbes in there will live in the colon and belong to the colon microbiota. Most of those are bacteria. You also have some archaea and some yeasts. Most of the archaea would be methanogens, especially in the genus Methanobrevibacter. For the yeast, or for the fungi, most of them are yeasts belonging to the genus Candida. For the bacteria, that's where most of your diversity is going to be. The 2 main phyla that you have in your colon would be Bacteroidetes and the Firmicutes. Mostly the Bacteroidetes, and then a lesser amount of Firmicutes. Actually, in the pie chart there, you can see the breakdown of the bacteria in the colon and how they're classified. So this big pie chart is showing the phyla. So it's mostly Bacteroidetes, and then a good amount of Firmicutes, and then small amounts of other phyla. Then if you zoom in on the Bacteroidetes, it shows the different families of Bacteroidetes that are found in the colon. And if you zoom in on the Firmicutes, it shows the different families of Firmicutes that are found in the colon. For the Bacteroidetes phylum, the largest family, or the most common family to be found in the colon, is Bacteroidaceae. Then for the Firmicutes, there's 2 families that are commonly found in the colon, which would be Lachnospiraceae and Ruminococcaceae. You always know you're looking at a family name because it ends with the aceae. Most of those bacteria would be doing some type of fermentation for their metabolism, although there's some exceptions. E. coli is one of the bacteria that's commonly found in the colon. That's actually facultative aerobe, not a fermenter. There's a good amount of diversity between individuals in terms of the makeup of their colon microbiota. Again, there's less diversity within a family, so in general you would share a lot of the species with people who you're closely related to and people you grew up with, people you share a diet with. But if you're looking between families, there's a whole lot of diversity. And also, of course, across the world, a lot of diversity. There's actually so many microbes in the colon that it accounts for a large part of the weight of feces. Actually, about a third of the weight of feces is just the bodies of the microbes from the microbiota. So you're constantly losing large amounts of those microbes in your feces, and constantly new ones are being born as the microbes that are left behind in the colon continue to metabolize and reproduce. So it's really a massive amount of microbes in the colon. Since there are so many microbes in the colon, that's kind of been a focus for a lot of research studies on the microbiome. So the microbiome of the gut, or the microbiota of the gut, is kind of better studied than the microbiota of other parts of the body. And as I mentioned, there's a lot of diversity between individuals in terms of the makeup of their colon microbiota, but studies have found certain, similarities or certain types of microbial communities that have been called enterotypes. And so far we've identified 3. So the hypothesis would be that everybody has, even though there's diversity in their microbiota for the gut, they're going to have 1 out of 3 types of basic communities there. So they'll either belong to enterotype 1 or 2 or 3. In enterotype 1, it's going to be dominated by the genus Bacteroides. So that would be a genus that is, much more commonly found in their colon than would be found in the colon of a person from a different enterotype. Then along with that defining genus of Bacteroides, you're going to be more likely to find certain other genuses or genera and less likely to find other genera. For enterotype 2, the dominant genus would be Prevotella, and for enterotype 3, the dominant genus is Ruminococcus. In the diagram there, it's kind of showing sort of maps of the different enterotypes and what types of genera you would find in those enterotypes. So this first little cluster is for enterotype 1, and in the red circle you have Bacteroides. So that's the dominant genus for that enterotype, or the defining genus for that enterotype. And then you have in the white circles other genera, or in some cases orders, like Clostridialis would be an order, not a genus. If you have a yellow line connecting Bacteroides to those other genera, then you're not likely to find that genus in a person who has enterotype 1. So if you have a lot of Bacteroides in your gut community, you're less likely to have Geobacter also. If the other genera are connected by a blue line to Bacteroides, then you are more likely to have them. So if you're enterotype 1 and you have a lot of Bacteroides, you are less likely to have Geobacter, but you're more likely to have Parabacteroides. And you're more likely to have order Clostridiales in your gut microbiota. And it's the same for these others. This would be enterotype 2 with Prevotella as the defining genus, and enterotype 3 with Ruminococcus as the defining genus. So if you have Ruminococcus, you're likely to have Staphylococcus also in your gut microbiome. You're not likely to have Sphingobacterium. If you have Prevotella, if you're enterotype 2 and you have a lot of Prevotella in your gut microbiota, you're likely to have Desulfovibrio in there as well. You're actually less likely to have Escherichia, which would be like E. coli. So this is still, this is still a hypothetical. So the enterotypes haven't been demonstrated with enough, evidence to say for sure everybody belongs to one of these 3 enterotypes. It's just kind of, us beginning to wrap our heads around the types of microbial communities that can exist in the gut. And so far, we're seeing that these 3 types are commonly found. And we haven't found other common types as yet. It is important which enterotype you have. Different enterotypes will have different metabolisms that they can conduct. They would probably have different benefits for you or perhaps different drawbacks for you depending on which enterotype you have. Some enterotypes might be more associated with a particular disease or less associated with a particular disease. So if we're able to understand these enterotypes better, then we might be able to give people kind of personalized treatments for certain gut diseases depending on what enterotype they have. Also, it could let us know like how they would respond to different types of treatment for certain gut conditions. So the gut microbiome can have a lot of different functions. One of them would be to digest fiber. So fiber is not digestible to you, but certain members of the microbiome in your gut are able to digest it and break it up into smaller pieces that you are able to digest. So that way you're able to get some nutrients from that fiber instead of it just passing right through you. You also have members of the microbiome that can make things for you that you cannot make yourself, but that you need, like vitamins or essential amino acids. So both vitamins and essential amino acids are, things that you have to have in order to live, but you can't make them yourself. Your body cannot synthesize them. But some members of the microbiome in your gut can synthesize them. So you have microbiome members that can make vitamin B12 and vitamin K. There's also a lot of other vitamins that could be made by the gut microbiome. Those are just kind of the most common ones. Especially other B vitamins are made by different microbiome members. And then the essential amino acids, they're just, amino acids that you have to build your proteins, but that you can't make yourself. So some microbiomes can make them for you. Microbiome members can make them for you. They also do some things that aren't necessarily positive, but could be positive, but could also be maybe negative. For instance, metabolizing steroids to a different form of the steroid, and then that gets absorbed back into your body. So steroids would be made as hormones by different organs of the body. Commonly the adrenal gland, which makes the stress hormones, which are steroid hormones. So in this diagram here, that kind of shows that happening, where you have the adrenal gland. When you get stressed out, it's going to release your stress hormones, which include corticosterone and cortisol. Those are both steroid hormones. So those are going to do different things that help you respond to stress, like increase your blood sugar, increase your blood pressure, increase your heart rate. Assuming that this stress is kind of the type of stress that you would need to fight or run away from, those things would help you. Some of those steroids are going to end up going through the liver also, and in the liver they're going to be metabolized to different products and excreted in the bile. Bile is made by the liver and it's excreted into the small intestine to help you digest your food. But now these steroids are also being excreted into the bile, so they end up in the small intestine now. Then they go into the large intestine, where you have microbes that are able to convert them into a bunch of different things. So corticosterone can be converted into different types of progesterone by microbes in the large intestine, and then that can be absorbed back into the blood. Cortisol can be converted to all of these different steroids by microbes in your large intestine. Some of these are testosterone analogs, and then those can be absorbed back into the blood, and they're all going to do different things. A lot of the time they do things that are similar to what corticosterone and cortisol would be doing, for instance, increasing your blood pressure and your heart rate, but they might have a different potency. So you might end up with some of your corticosterone being converted into something that's not as potent, and then you have a smaller blood pressure increase, which would probably be good. Or you might have something like cortisol being converted into some other steroid that is more potent, and then you have a bigger increase in your blood pressure and your heart rate in response to stress. So that would not be good for you probably. So that can be either good or bad, just depends on the exact microbes that you have in your gut, and we don't really understand the full implications of that yet. Another thing we don't fully understand yet is the gut-brain axis, which is kind of the connection between the gut and your brain. So that includes, the neural connection with actual neurons traveling from the gut to the brain and back. But it also includes the microbiome or the microbiota of the gut, since you have microbes in that microbiota that are able to make compounds that actually can cross the blood-brain barrier and affect the brain. We've actually observed mice having their behavior altered by products that were produced by the microbes in their guts. So we really don't understand this at all yet, really, other than it does exist. It could potentially be a good thing or a bad thing for sure, but it does appear that the microbiome in your gut has some ability to affect your behavior and change the way that your brain functions. Then finally, the gut microbiota has a large role to play in immunity. It has a lot of functions that are related to the immune system and preventing infections and disease. So it's really important for the development of your immune system. So out of all the microbiota that you have on the different parts of your body, specifically is the gut microbiota that is most important for the development of your immune system. You have to have a healthy microbiota in your gut early in your life in order for the immune system to develop properly and to learn the difference between you and foreign things. So actually the immune system in your early, childhood as an infant, your immune system should be learning that the microbes in your gut microbiota are actually part of you. It should be identifying them as self. That way it doesn't react to them later on. If that doesn't happen, if your immune system does not learn that those microbes are part of you and are not foreign, then later in life, when the immune system is exposed to those microbes, it will attack them. That means that it's going to be attacking your own gut, which leads to disease, of course. So if you have a poor microbiome in your very early life, that's more likely that your immune system will not develop properly and that later on it will end up identifying microbes that belong to your own microbiome as being foreign and hostile and attacking them and therefore you. Another thing that increases that risk is using antibiotics early in life. So in general, it is actually a good thing for infants to be exposed to some amount of microbes so that their immune system can learn, that their microbiota belongs to them. If you're using antibiotics in very young babies, then the consequences can be that you basically wipe out their gut microbiota. All those microbes die, or most of them die, and then the immune system has no microbes there to help it learn, that those microbes would belong to this infant and to this individual. So the more antibiotics are applied early in life, the greater the chances that the immune system will not learn that the gut microbiota belongs to that individual, and then that will cause problems later in life, or can cause problems later in life. So in addition to the gut microbiota being essential for the immune system developing correctly, you also have other ways that it helps you prevent disease. So some microbes in your microbiome or in your gut microbiota actually make compounds that are able to help your immune system find pathogens if they do come through your gut. You also have some microbes in the microbiota for the gut that make substances that are actually antimicrobial chemicals that prevent growth of other microbes. So they would actually be making those things in order to prevent their competitors from growing too fast, so that they can eat the resources instead of their competitors doing it. But for you, it actually helps prevent pathogens from growing if they get into the gut. And then finally, just basic competition. All of the beneficial or at least harmless microbes that live in your gut microbiota will be competing for resources with any pathogens that come through, which means that the pathogens will probably never be able to get enough resources to really have like a population explosion that could lead to disease. So it's actually not crazy uncommon for somebody to have pathogens in their gut microbiota or organisms that could cause disease. But these pathogens are just present in really low numbers, and the numbers are just too low for it to ever actually cause disease. And the numbers would be kept low because of the beneficial members of the microbiome that are competing with them for resources, and in some cases producing antimicrobials that prevent them from growing, or even producing substances that help the immune system identify that pathogen and kill it. The next organ we'll talk about that has a microbiota is the mouth. So that's called the oral microbiota. Technically, the mouth is actually part of the gastrointestinal tract, but we won't worry about that. There's a lot of different places in the mouth where microbes can live. You have a lot of different microenvironments in the mouth. Each would have its own conditions, so each is going to have its own microbiota. So first you have the saliva, of course the liquid that fills the mouth. There's actually not very many microbes in the saliva. It's not a very nice place to live. It actually has antimicrobial compounds in it, and it doesn't have very many nutrients. So there's just not very many microbes that survive in the saliva. Then apart from that, you have all the different surfaces of the mouth, which there's a lot of different surfaces. So you have the teeth, of course. The teeth above the gum line is actually a different microenvironment from the teeth below the gum line. So you have different microbiota for each of those microenvironments. Then you have the gums themselves, the tongue, different surfaces on the tongue, the hard palate, the soft palate. All the different surfaces in the mouth are going to have basically their own microenvironment and their own microbiota. Most of those are going to be oxic environments, so there's oxygen present, and you would have aerobes or at least anaerobes that can tolerate oxygen. But the little crannies are going to be anoxic, so like the teeth below your gum line, there's no oxygen that gets down in there, so that's an anoxic environment where you could have anaerobes living. Most of the microbes in the mouth would be living as part of a biofilm. So the plaque that grows on your teeth every day, that's actually a biofilm, and you clear it off. Also, all the other surfaces in the mouth are able to form biofilms. So that's why every now and again it's a good idea to brush your tongue too. Actually, most of those microbes in the mouth are facultative aerobes, so they can tolerate oxygen or survive without it. Well, I should say they can use it if it's there, or they can survive without it. And a lot of them are beneficial for you. The main thing that they would be doing to help you is just preventing pathogens from getting a foothold in the mouth. So if you have beneficial species in your oral microbiota, and then a pathogen comes along that would be able to cause gum disease or damage your teeth, it'll be harder for that pathogen to grow to large levels because it has to compete with all the other beneficial or at least harmless microbes that live in your mouth. So the diagram here, the pie chart is showing again the breakdown of different types of bacteria that are found in the mouth. The big pie chart has the phyla, the major phyla that are found in the mouth, in the microbiota of the mouth. So you have a lot of Bacteroidetes and a lot of Firmicutes, but you also have actinobacteria and a good amount of Proteobacteria and even a good amount of Fusobacteria. Then for each of those four major phyla in the mouth, the Bacteroidetes, Actinobacteria, Firmicutes, and Proteobacteria, you have a smaller pie chart that's showing the different families that are found in there. So for Bacteroidetes, here's the little pie chart showing the different families of Bacteroidetes that are commonly found in the mouth. Bacteroidetes is also really commonly found in the gut, but when you look at the family level, it's actually different. In the gut, you have a lot of Bacteroideaceae. In the mouth, you would have Prevotalaceae, Flavobacteriaceae, and Porphyromonaceae. You also have microbes that live in the respiratory tract. The respiratory tract is divided into the upper respiratory tract and the lower respiratory tract. The upper respiratory tract extends from the nose down to the larynx, which is in your throat. That's where your airway branches off of the esophagus in your throat. And then the larynx would also be housing your voice box. Then you have the lower respiratory tract that begins below the larynx at the trachea and goes down through the lungs into the alveoli. So in the upper respiratory tract, you have a microbiota, and you do not have a microbiota in the lower respiratory tract, or at least you should not. There should not be any microbes just living in the lower respiratory tract in your lungs. In the upper respiratory tract, though, you can have microbes that live there. So the entirety of the upper respiratory tract is going to be lined with mucus, I guess except for the vocal cords themselves. That mucus is designed to trap any particles or microbes that come in the air that you're breathing in. So any microbes that come in are going to get stuck in that mucus, and the microbiome members are able to actually live in that mucus and prosper there. Usually, though, they're not going to stay there for all that long because that mucus is always being moved. The cells in the the cells that line the upper respiratory tract, not only do they make mucus, but they also have cilia, like little whips that move the cells, that move the mucus. Sorry. So you have pathogens come in, or particles come in, beneficial microbes or harmless microbes that come in, they get stuck in the mucus. The cilia is going to be moving that mucus along, moving it usually back further into the respiratory tract until it gets to kind of, the throat, or the pharynx would be the fancy name for the throat, and there you can swallow it. Sending all those microbes down to the stomach to die in the acid. So that's how things are kind of supposed to go. You have some microbes that are going to be kind of left over in the mucus, the bits of the mucus that didn't get swallowed yet. In general, it's nothing bad for you. It would generally be good for you to have those microbes there, actually, because if a pathogen does come through, like Staphylococcus aureus, then it would just be kind of hard for that S. aureus to gain a foothold and reproduce to large numbers when you have all these other microbiome members that are competing for resources with that S. aureus. Then in your lower respiratory tract, you hopefully should not have any microbes in there. Any microbes that came in on the air that you inhaled, they should have been stuck in the mucus, in the upper respiratory tract. Some of them might actually end up getting all the way down to the trachea, especially if you've been breathing through your mouth. In that case, the trachea is also lined with mucus, so they'll get stuck there, and they should get moved up and out by the cilia that line the trachea. They'd be just pumping that mucus up the trachea to the throat where you can swallow it. So you shouldn't have any microbes that persist there in the lower respiratory tract. And the diagram is just showing the upper versus the lower respiratory tract. Upper on the top goes all the way through the larynx, and the lower on the bottom, which begins below the larynx at the trachea and continues into the lungs. So you would have the microbiota in the upper part, in the nasal cavity, which is this whole region. Then you have the pharynx that starts behind that, which is divided into 3 sections, but you don't need to know that. So this is all the pharynx, basically the throat, and then that goes down to the larynx. Next you have the urogenital tract, which is called that because in men the urethra would carry both urine and semen. But then of course the urethra continues on up into the body and transitions into the bladder, and then from the bladder you have the ureter that leads to the kidney. So from the kidney through the ureter to the bladder, you should not have any microbes living in those organisms. They should be sterile. But the urethra, which is the passage that it actually opens to the external world and carries urine from the bladder out, that actually will have a microbiota. Most of those microbes that live in the urethra would live at the distal end, which is the end where it's going to exit the body. So the end that's right by the bladder shouldn't have very many microbes living in it. Most of those microbes in the urethra are facultative aerobes, so they can use oxygen if it is present. It would be present sometimes, potentially, but in general the urethra is mostly anoxic. Sometimes you can have pathogens in the microbiota for the urethra. That's usually there wouldn't be pathogens, but occasionally, including P. mirabilis. So P. mirabilis, which is Proteus mirabilis, is unusual out of the pathogens that could be found in the urethra because it's able to actually use the urea that's in urine to produce ammonia. So if you have P. mirabilis in the microbiota of urethra, of your urethra is going to be producing ammonia. And ammonia is actually alkaline, so that's going to raise the pH of the urine up to be, basic. And whenever the urine takes on a basic pH, it's more likely for kidney stones to form. So not only can P. mirabilis cause a UTI, or a urinary tract infection, but it could also encourage the formation of kidney stones, which are very painful. And then the figure here is just showing the urethra in men versus women. So here it is in women. Then that leads into the bladder. That should be microbe-free. And in men, their urethra is longer, which means that they're less likely to have microbes getting all the way up in there and causing a UTI. Actually, it would still consider, actually all of that is the urethra in men, all the way up to the bladder. Then rounding out the urogenital tract for women is the vagina, which also has a microbiota. The vagina is unusual because it's also an acidic organ, similar to the stomach. The difference would be, of course, that the stomach is like extremely acidic, and the vagina is only a little bit acidic with the pH around 5. The reason why the vagina is acidic is because of microbiome members in its microbiota, specifically the genus Lactobacillus. So Lactobacillus is like by far the most common bacteria to find in the vagina. All of those species are doing lactic acid fermentation. The vagina will actually produce glycogen specifically for these bacteria, so they can use it as an energy source for lactic acid fermentation and convert it to lactic acid. And then the lactic acid creates an acidic environment in the vagina. Having that acidity is important for health because it discourages the growth of pathogens, just helps prevent disease. Different people will have different species in the Lactobacillus genus in their vagina microbiota, and exactly which species you have dominating is actually going to determine what the pH is. So different species would actually produce like different amounts of lactic acid and lead to a different pH in the vagina. It's also, I guess, unusual in that the Lactobacillus genus is only going to be common in the vagina after puberty, but before menopause. So before puberty or after menopause, you will not have the Lactobacillus, or you would lose that genus from the microbiota, and then you're going to lose your lactic acid fermentation, which means there's not going to be the lactic acid, which means the pH would go back to neutral. Again, making you in general more vulnerable to infections. So the figure there, or the graph, the pie chart, is just showing the makeup of the vaginal microbiota. So the big pie chart is the phyla, and you can see it's almost all phylum Firmicutes. Then if you zoom in on the Firmicutes to see the families, you can see it's mostly Lactobacilliaceae, which would be the family that has Lactobacillus. If you zoomed in even further to the genus level, you would see it's mostly Lactobacillus. The last organ we'll talk about that has a microbiota is the skin. So the skin's a very large organ, and it actually has a lot of different microenvironments because it's not the same all over your body. So some places you have hair, or different amounts, different types of hair. Some areas of the skin have more oil glands or more sweat glands. And of course you have places where there's folds and stuff that cause different conditions to develop. So different areas of the skin would have different temperatures, different moisture levels, different pH levels, different oil levels, different salt levels. And salt has salts in it, so different salt levels, and sweat also has antimicrobial chemicals in it. So if you're talking about a fold of the skin, for sure it's more likely to be warm there and moist there. And other parts of your skin would be cooler and drier. Then you also have pores all over the skin where microbes could live down inside the pores. And the pores are going to be anoxic environments. So in general, of course, the whole skin is exposed to oxygen, but if you're at the bottom of a little pore, oxygen might not make it down that far. Then in addition to that, you have other things that affect the environment on the skin, such as just the weather. Is it humid? Is it hot? Did you walk through a rainstorm? What gender you are also affects it, partly because your gender affects how much oil you produce, and exactly what the composition of that oil is. So men would have more protein in their skin oil, which makes it kind of more nutritious for microbes. Your age would also impact it. Your hygiene, so how many times you're washing those microbes off of your body in a given time period. And what pets you have. Since you pet your pets a lot, probably, you transfer their microbiome to you. So in addition to that, just things that you touch commonly can affect your skin microbiota as well. The microbiota of the skin is very diverse. You have, of course, a lot of bacteria in there, but you also have archaea and fungi, especially candida, but other fungi could be in there as well. There's three genera of bacteria that usually dominate the microbiota for the skin. So you have Corynebacterium, Propionibacterium, and Staphylococcus that are all quite common to find in different areas or different microenvironments on the skin. If you have moist skin, you would usually find a lot of Corynebacterium and Staphylococcus living there. If you have oily skin, you would have more Propionibacterium. In general, Propionibacterium doesn't like to be exposed to oxygen also, so that's common to find living like deep in pores, and more common to find living there if the skin is very oily, because it actually would be eating that oil. Then the Propionibacterium actually can cause acne, which is not in general serious, but we don't like it. And then on dry skin, it's kind of more of an equal mix of Corynebacterium, Staphylococcus, and Propionibacterium, with no one really dominating. See, in these graphs down here, the pie chart is showing the different phyla that are commonly found in the skin microbiota. So a lot of actinobacteria, a lot of firmicutes, a lot of proteobacteria, a lot of bacteroidetes, and a little bit of a lot of other things. And then here you have kind of the amounts of each of those phyla that are found in different areas of the skin. So sebaceous skin, that's oily skin, then moist skin and dry skin. So if you have sebaceous skin, that's going to be a lot of Propionibacteria, and then a bit of Staphylococcus and Corynebacteria, then some beta proteobacteria, and a lot of other. If you're talking about moist skin, though, that's going to be a lot of Corynebacteria and not so many Propionibacteria. A lot of Staphylococcus and a lot of beta proteobacteria. And then finally, the last figure is showing kind of a heat map for different parts of the skin where Staphylococcus would be found for men and for women. So on top you have men, and on the bottom you have women. And they basically sampled from different parts of the people's skin all over their bodies and saw how much Staphylococcus was growing in that area of their skin. So blue means not a lot of Staphylococcus. White means, maybe a medium amount, and red means like a lot. So you can see that for both men and women, most of the staph on our skin is living on our feet, especially the bottom of the feet. So we had mentioned that the microbiome, especially the gut microbiota, has connections to health and disease, and that studying it is kind of hard. But it is important that we continue to study this, especially with the gut microbiota, because there is clearly a large connection between your gut microbiota and different diseases, including cardiovascular disease and cancer and diabetes and obesity, although obesity is not actually a disease. In general, it's hard to study the links between the gut microbiota and these different diseases. Partly because, like I had mentioned, you can't ethically do things that would harm, a person who's a research subject. But also partly because the gut microbiota is influenced by a lot of different things. Of course, your diet is going to influence it, but also other things you might not think about, like your exercise level, it will influence it, and your genetic background and family history can influence your gut microbiota. So if you're just doing a study where you're kind of sequencing a sample from the gut microbiota and looking at the rRNA there, that's not really telling you as much as you would like, because you don't know anything about that person's diet or their genetic background, or just their lifestyle. So new studies in human subjects that are looking at the gut microbiota would, in addition to taking samples and sequencing the rRNA in those samples, you would also have the person tell you like what types of things they eat, like what their diet consists of, and what their exercise levels look like and stuff like that. So you would be tracking their diet over time and tracking their microbiota over time to get a lot more information. And then of course tracking their disease development over time as well. The issue with that is that at best you are only going to get correlational information out of it. That type of study will never prove that a certain type of gut microbiota is linked or causes a certain disease. It could only prove that it's linked to a certain disease. Right, so if you see that people who have a certain microbiota are getting cardiovascular disease all the time, that will let you know that there's an association there, but it does not let you know that their microbiome is what caused their cardiovascular disease. In order to prove that the microbiome caused the disease, you would need to take someone who's healthy, give them that microbiome that you think causes the disease, and then watch them get the disease, which is completely unethical and cannot be done in human beings. So in order to get that type of information, you have to use animal models, and usually they use a mouse model. So you can have mice that are healthy or have different health conditions, and you introduce certain species into their gut microbiota and just kind of watch what happens to them to see if certain microbiome members might cause disease, or if they might prevent disease, or I guess delay disease progress. So that's really useful. The only issue there is that, mice are not people. So you can get good information that way, but it's not going to translate just 100% to human beings. Their guts work differently than our guts do, and their microbiomes are different from our microbiomes. So overall, this is a really hard thing to study, but studying it is very important. And if we keep linking away, then we'll keep getting more and more information, and eventually we will be able to figure it out, even though we can't do, experiments that that would give us the most information. So we'll talk a little bit about how the microbiota in the gut develops during life, or changes during life. So when you're a fetus, you should not have a gut microbiota. It should be sterile inside of there. Then when you're born, that's when your gut is first colonized by microbes. So first, if you were born in a traditional pregnancy, then the infant would be colonized by the microbes from the mother's vaginal microbiota. If you're born by a C-section, then of course that's not going to happen. So in general, for babies that are born from like normal childbirth, you're going to see a much closer relationship between their gut microbiota and their mother's, because their mother's microbes are what had first colonized them. For a C-section birth, there's fewer similarities between the mother's microbiota and the baby's. Then apart from that, the other first colonizers for the baby's gut would be coming from their diet, so either formula or breast milk, and then also from people who touch them frequently. So of course, babies are always like putting their hands in their mouths, so if you touch the baby, then your microbes got onto their hand, and then they put their hand in their mouth, so now it's in their mouth, and they're going to swallow it. And if it survives the journey through the stomach, then it may colonize their gut. So people who touch the baby very frequently would be able to, have their microbes colonize the baby's gut. If you just touch the baby one time and never again, that's, it's probably not going to do it. So that's the reason why an infant's microbiome would look similar to the microbiome of people that they share a residence with, because those are the people that are probably going to be touching them and interacting with them the most frequently. And then, of course, diet plays a large role as well. So after you've got that first initial microbiome established with all those first colonizers that have come from possibly the mother's vagina, possibly this, and otherwise, definitely coming from their diet and from, people who have touched them. After you've got those first colonizers in place, then the development of the microbiota for this baby would depend mostly on their diet. And we see sort of characteristic differences between the early gut microbiota of a baby who had been breastfed versus one that was formula fed. So in general, breastfed infants would have a microbiota that looks in their gut maybe a bit healthier. It's going to be more diverse with fewer pathogens in it, and just kind of develops more rapidly to look more like an adult microbiome or microbiota. So in the future, if we are able to really understand the gut microbiota a lot better, then we could actually make formula that would be essentially kind of like probiotics, and that would have specific species that we know are associated with health and will promote the development of a healthy microbiota. Then the next big event that's going to impact the developing microbiota in the gut would just be when the infant switches to eating solid food. And at that point, their microbiota is going to start looking more and more like an adult microbiota. And by the time they're about 3 years old, between 1 and 3, depending on when they've switched to solid food, then they should have basically a gut microbiota that looks like what an adult would have. For an adult in that country who would be eating, a similar diet. So in this graph here, or diagram, it's kind of showing a summary of the development of a gut microbiota. So as the infant is just born, the microbiota would be first established from, in general, the mother's vagina or her skin. If it was a vaginal birth, then you would have more microbes that had come from the vaginal microbiota, especially a lactobacillus. If it was a C-section birth, then you would have fewer of those microbes from the vaginal microbiota, and more Staphylococcus, Corynebacterium, and Propionibacterium, which would be the dominant skin microbiota bacteria. And then after that, apart from just the skin of people who are touching them, the microbiota would be influenced by diet. So if the baby is eating a diet of breast milk, then breast milk is actually going to have some microbes in there, including Bifidobacterium, which is actually beneficial for the baby's development. So then the microbiome would have a lot of Bifidobacterium in it. If the baby is eating formula milk instead, then the microbiome would be different. It would have more Bacteroides, less Bifidobacterium, more Clostridium, and in particular more Clostridium difficile, which is a pathogen that can cause really serious diarrhea. So that's not a good thing. Then if you had also applied antibiotics, if the baby had needed antibiotics during that time period when their microbiome is first being established, that's going to have really significant effects on it. It's going to increase Enterococcus species, decrease the beneficial Bifidobacterium, and increase Proteobacteria, just leading to overall an altered microbiome. Then when the baby starts moving to solid food, then the microbiome will be changing a lot and becoming a lot more diverse. A lot of diversity introduced by that diet. And you see some of the early Bifidobacterium species, those are kind of dying out and becoming less common as new species enter. And then once the baby has basically moved to the solid food diet, then their microbiome would look more like an adult microbiome, and now they're actually not a baby anymore. So in that case, the microbiome would be determined mostly by their diet, which is going to depend on, as what culture they're growing up in and stuff like that. Just, what diet the family is eating. If this is more of a traditional diet, then the microbiome might look more like this, with a lot of Prevotella in it. If it's a Western diet, then it'll look more like this, with a lot of Bacteroides in it. In general, so I'm not, specifically sure if one of these microbiomes is better than the other, the traditional food versus the Western food microbiota, but in general, the Western food diet is associated with cardiovascular disease and diabetes, whereas the traditional diet is not. So it's definitely it's an unhealthy diet, that's for sure. So once you've transitioned to solid foods and your kind of adult gut microbiota is established, at that point you are not an adult, you are still a child, but you do have an adult gut microbiota, and that will persist until your old age. There's probably going to be around 200 species in there at the most, or maybe less, and most of them will be very stable over time. So there's some species that might be there, for a little bit of time, and then they'll kind of die out and maybe be reintroduced later, depending on what you're eating. But most of the species in your gut microbiota would just always be there and will be stable throughout your life. So those stable species that are always there are called part of the core gut microbiota. Because they're the ones that don't change. Those are also the ones that are more likely to be shared with your relatives. The ones that kind of are transient and just come in and come out, those are less likely to be shared with your family members. In particular, species that belong to the phyla Bacteroidetes and Actinobacteria are likely to be stable species that are part of the core gut microbiota. Species from the phyla Firmicutes and Proteobacteria are usually less likely to be stable species, less likely to be part of the core gut microbiota, less likely to be from a family member, more likely to be kind of in and out. Then when you are entering old age, the microbiome will start changing again. As you become older and older, it'll change, more and more. And in general, it's becoming less diverse. You would also be gaining overall more Bacteroidetes and less Firmicutes. So I guess the proportion of species in the gut microbiota would change to favor Bacteroidetes and have fewer Firmicutes there. And as well, just overall less diversity. In general, it's not a good thing for there to be less diversity in your gut microbiome. We're not sure exactly what the effects of that are, but it does seem to be associated with general frailty and with living in a nursing home, which means that when we study the microbiota of older people, the ones who live in nursing homes or are more frail have less diversity in their gut microbiota. So again, that's a correlation. That's not a causal relationship. Or at least we don't know if that's a causal relationship. So we don't know, is it like your gut microbiome becomes less diverse and then you become frail and have to live in a nursing home? Or is it the other way around? After you become frail and have to live in a nursing home, then your gut microbiota becomes less diverse. So we don't know which way that relationship goes. But it does seem like maintaining diversity in the gut microbiota is probably a good thing to do if you're able to do it. Then the diagram is just showing kind of the difference between an adult gut microbiota and an old age gut microbiota. So you can see there's a lot less diversity in the old age microbiota, and also the gut walls here are quite different. So all this stuff in the regular adult microbiota, or in the gut walls for this regular adult person, this is mucus actually, that's lining the walls of their intestines. And for the person who's in their old age, you can see there's less mucus. So the tops of these cells here are not being covered by mucus like they normally should be. Which, that's not a good thing. As you age, your gut does function less well. So it's not absorbing nutrients as well, not digesting nutrients as well, not producing mucus as well. And that mucus does protect the gut, you know. So some of those microbes in your microbiota are fine to be there just as long as they don't like get through that mucus though. And then when you're in your old age and you don't have the mucus, then they can, and it's, you know, probably not good. So now we'll go through the different organs whose microbiota we talked about, and just talk about some diseases that are related to the microbiota that occur in those organs. So first, with the gut microbiota, a major disease that's related to the gut microbiota is inflammatory bowel disease, or IBD. IBD always involves chronic gut inflammation, which means that you have an inflammatory immune response happening in the gut that just doesn't end. It goes on for a long time. There's different forms that IBD can take. It also includes ulcerative colitis, which is when you get ulcers in your colon that can wear away, you know, the wall of the colon. And it includes Crohn's disease, which is where you have a lot of abdominal pain and diarrhea and stuff like that. Both those diseases are all based around this chronic gut inflammation that is the hallmark of inflammatory bowel disease, or IBD. So IBD is one of these diseases that has kind of been around for a long time, but we haven't really known like what is the cause of it, and we still don't know what causes it. But a really good idea of what causes it might be that it's what happens when the immune system never learned that the gut microbiome belongs to the self. So if the immune system does not learn that early in life, for instance, because you had a baby that had to take, you know, multiple courses of antibiotics when they were very young, then the hypothesis goes that after that, you know, your immune system never learned your gut microbiota as being part of you, and now can react to it. But you're fine at first. So as time passes, and you're eating a Western diet, well, the Western diet causes the mucus that lines the walls of your gut to thin out. So then if it gets really thin in certain places, then some of those microbes that are in your gut microbiota can contact the cells of the gut wall, and you have some immune cells in there. So now these immune cells are in contact with these microbes from the gut microbiota, and the immune cells never learned that those microbes are part of you. So they're going to start an immune response. So then you're going to have, you know, more immune cells moving in. They're going to be causing inflammation. They're going to be constantly reacting to these microbiome members, and you're going to have a lot of dangerous signaling molecules released by these immune cells that cause damage to the other cells around there, that further thin the mucus lining the gut wall and damage the wall itself of the gut. So you can end up with an ulcer or some other type of lesion in the gut wall, and just this war against nothing will basically never end. And you'll just always have this massive inflammation in the gut, which just causes problems of increasing severity as you age. So it's not for sure that is what's happening in inflammatory bowel disease. That is a strong possibility, though. There is some evidence to suggest that might be what happens. We just haven't clinched it yet. This diagram here is showing kind of a summary of that. The diagram has kind of a lot of detail on it. We're not going to get into all of this detail with the different parts of the immune system, but on the left you have a healthy adults with their microbiome, and here you would have some immune cells in the wall of their gut. This would be a blood vessel passing by at the bottom, carrying more immune cells. And this is an immune cell that's kind of sampling from the interior of the gut to see what's in there. So since this is a healthy person, the immune system is not reacting to the microbes in the microbiome of their gut. The immune system knows that those belong to the person, so everything is fine. On the right, you have a person whose immune system did not learn that the microbes in their gut belong to them, and that's where the problems come in. So now when this immune system cell is sampling from the microbiome, it's going to find something that is not familiar to it, that it thinks may be a pathogen, and it's going to activate and cause an immune response. So you're going to have all these immune cells in the area becoming activated. They're all releasing chemicals that then activate other immune cells. You have immune cells from the blood coming in. These guys here are the cells that, well, they're plasma cells, which are the cells that produce antibodies. So now you have antibodies around here, as well as inflammation just going on and never stopping. And then as that continues, the cells lining the gut wall will gradually begin to degrade, which causes a lesion of some kind, possibly an ulcer. And so if you look at their gut as a whole, you'll see these red streaks where they have damage to the wall of their gut caused by all of this inflammation. So again, that's one possibility of what could be causing IBD. One thing that is known about IBD and the gut microbiota that is for sure, that is just that the gut microbiota of people who have IBD is different from the gut microbiota of people who do not have IBD. So again, that might or might not be causal, but there is some association or some relationship between the gut microbiota and IBD for sure. Another disease state kind of that's related to the gut microbiota is obesity. So obesity is not a disease. It's not properly a disease state either, actually, but it is a disease promoting state. So if you are obese, you're more likely to get certain diseases like cardiovascular disease or type 2 diabetes, although it itself is not a disease. So it is thought that the gut microbiota has some relationship to obesity. One, I guess, hypothesis that has been explored in mice and has found to be basically correct is that you have microbes in the gut microbiota that are able to digest fiber. Some of them, when they digest fiber, will convert it into what's called volatile fatty acids, or VFAs. Just fatty acids that are able to evaporate, which makes them volatile. So your intestine can absorb those VFAs and use them as an energy source. So fiber itself you cannot use as an energy source, but these volatile fatty acids you can use as an energy source. And then just very simply, if you have more microbes in your gut microbiota that make VFAs from fiber, you're going to have more energy that you're able to derive from that same diet. So even though you might have the same diet as another person who doesn't have those microbes that convert fiber to VFAs, you would be getting more energy out of your diet than them because you're getting more of these VFAs. And then maybe that leads you to put on weight even though you're eating the same diet as the other person. So let's say this has not been demonstrated in people at all. This has been demonstrated in mice. So in mice, this kind of figure on the bottom is showing, oh god, that's not like an arrow, is it? Cool. First you have the gut microbiota for a lean mouse, and then for an obese mouse. So for the lean mouse, you have not very many methanogenic archaea in there. They would be shown in green, so you don't have too many of those guys. In the obese mouse, you have a lot of those methanogens. So the fermentation reaction that is actually producing VFAs from fiber is inhibited by molecular hydrogen, or H2, which is also produced from that fermentation. So that means that if you don't have anybody consuming that molecular hydrogen, then there's only so much of this fermentation that's going to occur. You're only going to make so much VFAs. If you're an obese mouse, then you have a lot of these methanogens in your gut microbiota. Those methanogens are actually consuming molecular hydrogen and converting it to methane. So now these mice are going to have less molecular hydrogen, which means they're going to end up having more of this fermentation and more VFAs produced. And then the mouse, the mouse's body will use those for energy and deposit all of the extra energy as fat. So it has been shown that if you take a lean mouse and introduce these methanogens into their gut microbiota, they will put on weight. So how does that relate to people? It's not entirely clear. One study that's been done was looking at twins, and that's what's shown in this other figure here. So you had an obese twin and a lean twin, but they are twins. So otherwise, they should have similar genetics, and they should have been exposed to basically the same environment. If you take a sample from their gut microbiota and deliver that sample into the gut of different mice that are, starting out as lean mice, and then wait and see what happens, feeding them, both the same diet, the mouse that got the transplant from the lean twin remains a lean mouse, and the mouse that got the transplant from the obese twin becomes an obese mouse. So it certainly seems that something in this microbiota transplant that the mouse received caused that mouse to become obese, even though the mouse was eating the exact same diet as the mouse that did not become obese. So it does seem to have a relationship to the microbiome. Is it just as simple as depending on what microbiome you have, that's what's going to determine, how much you weigh? No, it's not that simple at all. Obesity is an extremely complicated phenomena. It's being actively studied by scientists in a lot of different subfields, actually, because it's very, very complicated. So you have brain scientists that study obesity, you have endocrinologists that study obesity, you have, with the microbiota, you have microbiologists now that study obesity. All of these disciplines have, I guess, something to contribute to the understanding of obesity, because it's very, very complicated. So, for instance, obesity is always associated with inflammation. Does the inflammation cause the obesity, or does the obesity cause the inflammation? That we don't know. We just know that they always go together. Obesity is also associated with insulin resistance. Again, we don't know which one causes the other. Advertising and store design are designed, they're intended to make you eat food that you would not otherwise eat. Right? When you're watching an advertisement and you see an image of a delicious hamburger, you want it. Your brain doesn't know that it's not there. Your brain grew up or kind of evolved in an environment where food doesn't grow at the store. You have to go out and find it growing somewhere in the ground, or you have to hunt it. So if you see food, you should probably eat it, because there's not going to be food again for who knows how long. We don't live in that world anymore. Now food is everywhere all around us, and we're bombarded with images of it constantly, such as in advertising. Every time you see something like that, your brain is wired to make you want to eat it, because you are designed to live in an environment that you do not live in anymore. Stores, if you go into a grocery store, you always find like the sweet stuff at the front, right where you walk in, because who doesn't want some cupcakes? In any store that you're at, basically, when you're at the checkout line, there's candy bars and chips right there. Because they're trying to get you to make an impulse buy. You have to stay there in line. You're going to pause. You came to the store. You got whatever you wanted. Now you're paused at the checkout line, waiting to buy your stuff. And what is there for you to see? A wall of candy. In attractive coloring, like yellows and oranges and reds are shown to be associated with like increased food intake. Some people will pick up that candy bar and buy it, that they would not have gotten it otherwise if it wasn't right there put in front of their face. Those are deliberate tactics designed to manipulate your behavior to make money for someone else. But the result for you is that you're consuming calories that you would not otherwise consume, and that are in general not healthy calories to consume. You also have, and related to that, in the Western diet, a lot of highly processed, highly palatable foods, which means, they take the initial food, they strip all the nutrients out of it, and they pack it with fat, salt, and sugar, things that make it taste good. But nutritionally, it's empty. There's not vitamins in that. It's not good for you to eat it, but it tastes real good. You are evolutionarily wired to want the taste of fat and sugar. When your tongue detects sugar or fat, you increase the rate at which you eat. And that's not about your personality. That's about the fact that you're a human being who's designed to survive on a savanna where food doesn't come like with 70 grams of sugar in it. And you have to hunt for it first before you can eat it. Another thing that's related to these empty calorie foods that taste real good with all the fat and the salt and the sugar but aren't good for you, they don't have these vitamins and trace nutrients that you need. So one thing that can happen is that even though you're eating a lot of calories and you're gaining weight, you are still malnourished at the same time because you don't have the nutrients that your body needs to survive and to thrive. And it's possible that your body is aware that you're not getting some of these vitamins and stuff that you need, and so it makes you hungry because you need those vitamins. But then what do you go out and eat? It's the food that's around you, which is Western food. It's very processed. It's very palatable. It's not very nutritious. It doesn't have vitamins. So you're stuck in this cycle of trying to eat more food to get the nutrients that you require, but the food you're eating doesn't have them. It's just helping you put on calories. Then, in addition to all of that, of course, your exercise levels is going to affect your weight. Also, your genetics has a role to play. Some people are just genetically disposed to have a lean body type. Some people are genetically disposed to having a more obese or overweight body type. And more than that as well. That's just a small sample of the things that control obesity or relate to obesity in one way or another. Obesity is very, very, very complicated, you guys, is the point. The microbiota does play some type of a role, though. So then you have the oral microbiota. The two main diseases that would be associated with that microbiota would be cavities and gum disease. The fancy word for cavities is dental caries, and the fancy word for gum disease is periodontal disease. For dental caries or cavities, that's caused when you have kind of pathogenic bacteria or harmful bacteria living in the oral microbiota that grow on the teeth and form a biofilm in there called plaque. So of course you're actually going to have plaque all the time on your teeth, except for right after you're brushed. And plaque is not always made up of pathogenic species. Usually it would be species that don't harm you. But if you get some of those pathogenic species in there and they kind of gain a foothold, they would be living in the plaque. And a lot of them will be producing lactic acid or some other type of acid, usually by fermentation. And then that is going to reduce the pH right there by your teeth, and a lower pH is bad for the enamel of your teeth. So then the enamel will be damaged, and when it's worn away, then the microbes can kind of get to the softer parts of the tooth that are underneath that and begin eating it and damage the tooth. Then for periodontal disease, what happens there is that, again, you're going to have some type of pathogenic bacteria introduced into the oral microbiota, which instead of dying out is going to, get a foothold and develop a significant population there. That's kind of what's shown in this little diagram here. So you have in purple a pathogenic microbe that's been introduced to the normal community in the mouth. And as a result of the colonization by this pathogen, if you're going to end up with disease, you would need the whole microbiome to kind of be altered in dysbiosis. So this pathogen is going to cause the microbiome to shift from this kind of makeup, from the healthy makeup, to the dysbiotic makeup. Dysbiosis just means that your microbiome is out of balance. Basically just that. So it has fewer beneficial species and more harmful species if you're in dysbiosis. Then this dysbiosis is going to cause inflammation. White blood cells or immune cells are going to move into the area and begin releasing chemicals that cause inflammation. Inflammation generally is associated with tissue damage. So as the inflammation damages the tissue of the teeth and the gums, you're going to have some of those dead cells from your tissue are going to end up feeding the bacteria, which is going to cause more bacteria. That's going to cause more inflammation. So this is a feedback loop where the tissue damage is going to get worse and worse until you have diagnosable gum disease or diagnosable periodontal disease. There's a couple different forms that can take. Hopefully you just have the gingivitis, which is not as bad. But if you get periodontitis, that's related to significant damage to the gum and to the tooth next to it, and even to the bone of the gum. So all that actual tissue damage there is being caused by the inflammation, not by the bacteria themselves. Until, of course, they get to be eating at the interior of your tooth. Both those conditions, the dental caries and the periodontal disease, are associated with low diversity in your oral microbiota. So if you don't have very much diversity in there, then it's more likely that when a pathogen moves in, they'll be able to establish themselves. Then for the skin microbiota, there's actually a number of different skin conditions that could be caused by pathogens in your skin microbiota. Like a lot, actually, but we're only going to talk about acne, which again, the fancy name is acne vulgaris. That's caused by Propionibacterium acnes, which is actually very normal to have in your microbiome. Most people have this bacteria, P. acnes, in their microbiome. Not everybody, but most people. Different strains are, different. So some strains of P. acnes don't really cause acne. Some strains cause acne more frequently or more serious acne. But we really don't know why certain people would have acne in response to certain strains of P. acnes and other people would not. We do know that there is a relationship between skin oil and acne, where the more oil your skin produces, the more acne you're likely to get. And that's because the Propionibacteria are eating that skin oil. That's their food. So they like to live in your pores. They are anaerobes, so they don't like air. They have to live down in the pores where they can hide from the air, and then they eat your skin oil for their food. And if they multiply to a large level and your immune system reacts to them, then you're going to get acne forming as a result. So the diagram is just showing kind of some different, how acne progresses and some different forms that it can take. So it's going to form in your pores. A lot of the time with your hair follicles, there's going to be those skin oil glands next to the follicles producing skin oil. And then if you end up getting that pore clogged, the oil is going to build up there and form a comedone. At that point, that's not any type of infection. That's just a plug made of the kind of congealed oil and maybe some keratin from your skin. Then if you had the P. acnes bacteria move into this comedone, it's actually full of food for it. So at that point, you would have an actual pimple because your immune system is responding to it. So in green, you can see the bacteria eating the skin oil in that former comedone, and the little blue guys around would be the immune system cells that are responding to the bacteria and causing inflammation. Then, if I guess you have more of these bacteria growing and the inflammatory response gets worse, then, the pimple is going to get worse as well. In some cases, it may end up spreading beyond the initial pore that it was in if the tissue around that hair follicle becomes damaged. Sometimes it can even get bad enough that it leads to scarring. So this is in general, this is not a dangerous disease, but it can, if it gets serious, cause scarring, which, obviously people don't like that. So that's why we try to get rid of our acne, I guess. If we were able to understand exactly what it is about certain strains of P. acnes that makes the immune system react to them, then we could actually theoretically cure acne. So next we'll talk about the vaginal microbiota and disease. There's two kind of basic disease states that are associated with the vaginal microbiota. First is vaginosis and then vaginitis. Vaginosis is less serious, vaginitis is more serious. In either case, you have certain microbes in the microbiota growing to be like too large of a population. In general, something that's not lactobacillus. So in either of these conditions, you would have probably a reduction in the amount of lactobacillus that's in the microbiota and an increase in the population of some other type of microbe, which would be kind of an overgrowth of that microbe. And then you have the immune system responding. In vaginosis, you have a weak immune response, so kind of low-grade inflammation. In vaginitis, you have a strong immune response, with a lot of inflammation and probably also a larger overgrowth in whatever microbe is causing this condition. So a yeast infection is actually a type of vaginitis where you have less lactobacillus microbes in the microbiota, and the yeast are beginning to multiply and kind of take over, and that causes a strong immune response. And then that immune response is mostly what's responsible for the symptoms of the yeast infection. Actually, the vaginal microbiota is constantly being disrupted when you menstruate, also when you have intercourse, and it'll have a couple of different properties. If it's healthy, it will have either high resilience or high resistance or both. High resistance means that the microbiota doesn't change much in response to disruption. High resilience means that when it does change in response to disruption, it quickly goes back to normal. So if you have low resilience and low resistance, then that is a microbiota that is vulnerable to being disrupted, and it is routinely disrupted because of menstruation, and at some point you probably will end up having the lactobacillus being kind of displaced by some other type of species that would lead to vaginosis or vaginitis. And these graphs are just kind of showing the difference between resilience and resistance. So on the top you have the microbiota of a person with high resilience. Over time, this is the makeup of their microbiota over time. In red you have lactobacillus, and then other species or other genera are color-coded according to the key here. And on the x-axis you have time. These little dark red splotches are menstruation. So every time you have a menstruation, that's going to be a disruption to the microbiome. And you can see that this person's microbiome is changing in its composition with a lot of these disruptions. But every time it changes in its composition, it also goes back to normal pretty quickly. So within maybe a week or a couple of weeks, this person is always back to normal. So that is a high resilience microbiota. On the bottom, you have someone with a high resistance microbiota. So you can see that even though they're menstruating, usually their microbiota doesn't change. It usually remains just lactobacillus. So I guess there are a couple other things to talk about. First, antibiotics in the microbiome. So we had briefly discussed this already, but anytime that you're taking antibiotics, that is going to disrupt your microbiome, especially the microbiota in the gut, since, antibiotics are usually oral, so you'd be eating it, and the gut microbes would be the ones who are exposed to it first before it's even absorbed into your bloodstream. So usually, you take the antibiotic, it disrupts your microbiome, especially the microbiota in the gut. But after you finish taking the antibiotic, your microbiota should reestablish itself, and basically go back to normal. So it should have resilience and go back to normal after being disrupted. Sometimes it doesn't happen though. So whenever you are on this antibiotic and your microbiome is disrupted, that's representing like a window of opportunity for a pathogen to move in and grow to form a large population, especially if this pathogen is resistant to the antibiotic that you're taking. So a really common example of this would be Clostridium difficile or C. difficile. So actually a lot of people do have C. difficile in their microbiota, even though they're healthy. And it doesn't cause disease because it's only present at low or in low numbers. So the population of C. difficile would be small, and it needs to have you need to have a large population to actually get disease there. But C. difficile is commonly antibiotic resistant, and antibiotic resistance is spreading, in C. difficile strains. So as time goes by, it's just more and more likely that the C. difficile in your own body would be, resistant to antibiotics. So if you have that antibiotic resistant C. difficile in your gut microbiota and you take antibiotic drugs, then you can have the C. difficile having like a population explosion because all of its competition is now dead. And once you stop taking the antibiotics, it's kind of a question mark what's going to happen. Sometimes the normal microbiota will still, be able to grow back to the levels that they had been at, and they'll displace the C. difficile, and it will again be outcompeted by them. But sometimes that won't happen, and instead you just end up with a microbiota that's full of C. difficile that's resistant to antibiotics. And then that causes, serious diarrhea and sometimes death. So that can happen to anyone. It's more likely to happen to a young person, and it's extra more likely to happen to an old person. So it usually does happen to old people. And this graph is kind of showing that. So the red line is your normal microbiota in the gut. The the green line is your C. difficile population level. So when antibiotic is applied, all these normal microbes start to die, and the C. difficile now doesn't have competition and it's resistant to the antibiotic, so it starts to grow to a real high level. And then after you stop taking the antibiotic, then you see what happens. Usually, the normal microbiome will reestablish itself, but sometimes, and then, if the normal microbiome reestablishes itself, then C. difficile will shrink back down to a low level. But sometimes that doesn't happen, so in the dotted line that's showing like C. difficile basically just taking over the microbiota for the gut. So that can happen, at any age if you're taking antibiotics, or always it's going to disrupt your microbiome, that's for sure. But if you're taking antibiotics really early in your life, like usually in the first few months when your microbiome is just being established for the first time, then that is extra bad. That's going to have a negative effect on the development of your immune system that can possibly persist throughout the rest of your life. That is going to increase your risk for autoimmune diseases because the immune system never learned that your microbiome belongs to you, because it just wasn't, it was disrupted when your immune system was trying to learn that information. Also, it's been linked with increased risk of childhood obesity. Not really clear what the causality is there again, but it does seem that babies who have antibiotic therapy within the first few months of their life are more likely to be obese as children. So in general, it's just a good idea to avoid giving antibiotics to a child that young if it's at all possible. And sometimes it's real tense because your kid is sick and you want to make them better. So, you just want to get something that's going to make them better. But if they're not seriously sick, then there's not actually a reason to use antibiotics. That could actually, be bad for them later in life if you were to do that. Another thing that that we do now is fecal transplants. In a fecal transplant, you're basically taking feces from someone who is healthy, who ideally would have a healthy gut microbiota, and you're transplanting that feces into the large intestine of someone who does not have a healthy gut microbiota, and you're just hoping for this transplant to replace their microbiota, and give them something more healthy. So this is actually quite effective. In some cases, it can be used to treat inflammatory bowel disease and those drug-resistant C. difficile infections. So, if you were on antibiotics and as a result you had your microbiome replaced with C. difficile and it's resistant to antibiotics, one thing that could be done to treat that is just to give you a fecal transplant from someone who still has, their healthy gut microbiota. That's actually very, very successful. It's usually successful, about a 90% success rate. I'm not sure what the success rate is for treating inflammatory bowel disease, but it works sometimes and sometimes it doesn't work. It's possible it could also be used to help with other conditions, like the metabolic syndrome. Metabolic syndrome is kind of just a constellation of related symptoms. That's that is what you would have if you are obese and you have like systemic low-grade inflammation and you have insulin resistance and you have high blood pressure. So all those things usually do go kind of hand in hand. So a lot of people have metabolic syndrome. It's possible that transplanting a healthy gut microbiota into their gut might help reverse that condition, although that has not been demonstrated yet. So I guess the fecal transplant business is kind of a Wild West right now. It's really not particularly regulated. But in general, you should be using someone for a donor who has been screened, who's been through an actual screening process, so you can actually know for sure that they don't have some type of disease that they're going to pass to you in this transplant. You shouldn't just get it from just anybody. The donor should be screened by a doctor. And probably the best way to deliver the transplant is in a liquefied form and during a colonoscopy, because that way you're going to be able to get it kind of further up into the colon. You're already up in there if you're doing the colonoscopy. There's also other formats that it can take. So in the picture you have, this is the liquefied fecal transplant sample. And then this is kind of a dehydrated fecal transplant sample that is been put into capsules like as a pill. So that's a less effective way of delivering. It's a less effective delivery method. This other figure is just showing basically, I guess, the concept of what's supposed to be going on. So you have over here a healthy person and their gut microbiota. So you take a fecal transplant from them. Hopefully they were screened as a donor and you know for sure they don't have any type of disease that will be transmitted to you in this process. And that is a liquefied sample that's going to be delivered to you during your regularly scheduled colonoscopy. And here's I guess in this example, this is you with your inflamed colon because you had been on some antibiotics that destroyed your gut microbiota, and your antibiotic resistant C. difficile population exploded, and now your microbiota looks like that, and you're sick. So you get this transplant, and time passes. These new microbes that you got from the healthy donor have prospered, and now your microbiome is nice and healthy again, and you're happy, and you do not have any diseases. Hooray! Okay, so the last thing is probiotics and prebiotics. A probiotic is a sample of living microbes that you ingest to benefit your microbiome. Prebiotic is when you're ingesting nutrients that are designed to help good microbes grow. So they're similar. Both of them are designed to kind of increase the health of your microbiota, particularly in your gut. But one, the probiotic is actual microbes, and the prebiotic is just nutrients for microbes. For probiotics, usually species that are used would be in the genera Lactobacillus or Bifidobacterium, although sometimes you see other genera as well. When you're eating a probiotic, you have to kind of eat a lot, because all those microbes are going to be passing through your stomach. If they're trying to get to your colon, they have to go through the stomach, and most of them will die because of the stomach acid. But that doesn't mean just buy a supplement and eat the whole box, though. Don't do that. Don't do that. Just be aware that most of the microbes you're eating are going to die in your stomach before they get to where you had wanted them to go. Probiotics are not really regulated. They've not really been scientifically studied very much. It can include just regular foods like yogurt. That's a probiotic food, or like kimchi. Anything that's fermented basically is a probiotic, or kefir, or kombucha. Those are all probiotics. Then of course you have actual like supplements that are like capsules or whatever that have the microbes living in them. And again, none of this is really regulated. And of course, we don't really fully understand the gut microbiota like at all yet. So as we do more science, as our understanding of the microbiota improves, then we'll be able to have better probiotics, basically. Theoretically, we'd be able to identify, like, with your microbiome the way it is, if you would have just this one specific species added in, that would be real good for you. And for another person, maybe it's a different species that they need. So then you could have, probiotics that are actually tailored to you, your microbiome, and your health state. And of course, these probiotics that we envision would be regulated by the FDA because they would be, essentially medicines, which, would be a good thing. So, related to this, if you're trying to buy some probiotics and improve the health of your microbiome, just be careful out there because supplements are not regulated by the FDA and they actually don't have to, not have poisons in them. So actually, nobody has to verify that these supplements have what they say they have, and no one has to verify that they don't have poisons, essentially, in order for them to be sold. So they can literally just, say whatever on the package and put, like just vegetable glycerin in a capsule and sell that and call it a supplement. No one will stop them. Because it's not regulated by the FDA. So actually, when this has been looked at in studies, it's been found that a lot of the herbal supplements on the market do not have the herbs they say that they have. In one study, 4 out of every 5 bottles that they looked at did not have the herb that the supplement said it had. In another study, 5 out of 6 companies that they looked at were producing herbal supplement products that didn't have the herb that it said that it had. So, on average, most of them actually don't have the things that they say they have in there. If they do have it, you don't know what dose it is. Like, it might not be what they say. There's, nobody's forcing them to, actually have what they say they're going to have, so they probably don't have it. Also, a lot of these supplements will have kind of other things in them, things you might not want, such as prescription drugs or illegal drugs. Especially like sex enhancement supplements or weight loss supplements. Even if they're saying that's an all-natural sex enhancement supplement, a lot of those have some type of analog of Viagra. Weight loss supplements real commonly have amphetamines in them, which amphetamines are analogs of meth, which, is a dangerous street drug. For these Viagra analogs, Viagra itself not so dangerous, but this is not actual Viagra we're talking about. This is some type of analog, which means that its side effects are going to be different from Viagra and sometimes more serious than Viagra. It's just like, I don't know if you guys have heard of spice. Of course, marijuana is illegal. THC is the active compound that gets you high. So people have synthesized analogs of THC, and those analogs are sold as spice, which is legal, not legal to consume, but legal to sell, not for consumption. But of course, people would only buy it to consume it. And these analogs, they're not like THC. They're not like THC. They're much I mean, how different it is depends on the analog, but the ones that are more like THC are now all illegal themselves. So what we've got left is some pretty crazy stuff. It's not at all like marijuana, and the effects can be quite serious. So you can read stories about people just kind of basically waking up paralyzed because of this stuff, or people just kind of like passing out, not knowing where they are or who's with them. Crazy things can happen. So, it's an analog of THC, but it is not THC, and it is not safe like THC is. So just because you have an analog of Viagra, it does not mean that it is Viagra or is safe like Viagra is or anything like that. That would be my point. When you have these companies selling this stuff, with illegal drugs in it, or prescription drugs in it, and they get caught, there's a penalty, there's a fine that they pay, but the fine is not that much, and the potential profits they can make by selling Viagra and amphetamines are very large. So, most of these companies, maybe I shouldn't say most, but there's been a lot of instances where it's been found the same company is violating the same thing over and over and over again. So, they just pay the fine, they turn around, they make the same product, they get caught again, pay the fine again. It's just a cost of doing business for them. It doesn't impact their profit margin significantly enough to make them stop doing it. Another thing I want to note is just because it's natural doesn't mean it's safe. So, you know, probiotics would be natural, but that doesn't mean that they're safe. Also, for any other supplement that's herbal or all natural or anything like that, natural is not safe. Natural just means not made by men. For an example, tetrodotoxin is natural. Botulinum toxin is natural. These are two of the most potent toxins known to man. You know, just a tiny, tiny little dose of them can kill you. Tetrodotoxin is the toxin found in pufferfish. Botulinum toxin causes botulism. It's made by certain bacteria. You know, these are natural products, but they're very, very deadly. A lot of these herbs, you know, of course it's not like toxic like tetrodotoxin is, but that doesn't mean it's safe. Actually, it's quite common for people to get sick as a result of taking supplements, even to die, because a lot of these herbs are, you know, even though they're natural, they're not exactly safe. And of course, none of this is being regulated by the FDA, so these companies do not have to prove that an herb is safe in order to sell it, or that a probiotic is safe in order to sell it. All they have to do is, you know, be able to sell it and make money like that. That's it. You know, the herb that they are selling may become illegal if enough people die, and there's kind of an outcry about it. But then there's no one to stop them from just keeping on selling it, basically, because the fine they would pay is very small. So, all that said, you just be careful with the supplements. It is a dangerous business, buying supplements. It is safer than buying street drugs, but not that much safer than buying street drugs, to be honest with you. If you're going to buy supplements, there's a few things you want to look for on the bottle of that supplement, which is a label from USP, CL, or NSF. USP is US Pharmacopoeia, CL is Consumer Labs, and NSF is National Sanitation Foundation. So these are all third-party entities. Some of them are for-profit, some of them are not for-profit, and they test supplements just to make sure that it has what the label says it has, and to make sure that it does not have certain contaminants. So the picture there is showing those labels. So this is the NSF label, the USP label, and the CL label. So if you see any of those labels on the bottle of the supplement, that means that that company or that organization tested the supplement to make sure that it has what it says it has and it doesn't have other things in it. So those are the only supplements that I would bother buying, because otherwise you're just you're really taking a risk. At the very least, you'll probably waste your money on buying something that doesn't even have what you wanted. But bear in mind that just because it has that label on it doesn't mean that it's like safe, like for sure safe. It means that it doesn't have any of the common contaminants that these organizations test for. But there's so many different possible contaminants and different drugs that you could put in a supplement that it's impossible to test for all of them. So, it doesn't mean for sure it doesn't have any extra drugs added to it. It just means that it doesn't have common contaminants like heavy metals and stuff in it, and that it does indeed have whatever it said it's supposed to have on it. So if you're looking for probiotics or any other type of supplement, I wouldn't bother buying it if it doesn't have a label like that on it. The other option is just to, stick to your fermented foods, like your kefir and your yogurt and your kombucha and your kimchi. Those foods cannot be made without the microbes, without the probiotic microbes in them. It's impossible to make yogurt without the right microbes, and those microbes are actually, probiotic microbes. And, there's just not a reason to put amphetamines into yogurt, so I'm guessing no one does. And, that's just going to be a safer alternative.